Hello dear students, today I am going to deliver my first lecture and that is based upon our first chapter that is Recombinant DNA Technology. And topic of today's lecture is the basic introduction about Recombinant DNA Technology. So Recombinant DNA Technology, in short, we use the phrase RDT. RDT stands for Recombinant DNA Technology. It is basically a collection of techniques. RDT is basically discipline under the field of biotechnology, and it consists of various techniques, and the objective of all the technique taken together can be summarized into the following two categories. One is the construction. The primary aim of RDT is basically the construction of a special type of DNA that is called as recombinant DNA. In short, we can write it as rDNA. And this rDNA is created in vitro conditions. It means within artificial environment, within glasswares, within laboratory. So this is the first basic or primary objective of RDT, that is the construction of recombinant DNA molecule in vitro. And after the construction of recombinant DNA, the second important objective of RDT is the use of that rDNA somehow by using some strategy to generate some sort of human welfare. It means once you create rDNA molecule inside lab, now will you use that rDNA by using some experiment, by using some technique, by using some process to generate some kind of product, some kind of service, some kind of organism, some kind of change in the organism, so that we can do some sort of human welfare. For understanding the discipline of recombinant DNA technology completely, first we have to concentrate on this recombinant DNA. So what is recombinant DNA? Recombinant DNA is a DNA that has been created in vitro by joining together two or more DNA fragments or molecules, usually isolated from different sources or organisms. What the definition is saying, that recombinant DNA is a type of DNA which has been artificially created inside laboratory conditions. And how it has been created? It has been created by joining together two or more DNA fragments. It means we have isolated two or more DNA fragments from different organisms, and later on we are joining all the DNA fragments together to make a single DNA molecule, and that joining has to be done inside laboratory conditions. And that single, you can say that hybrid DNA molecule, which contains all the pieces of DNA isolated from different organisms, that hybrid DNA, that is called the recombinant DNA. In this photograph, it is a very simplified photograph of a typical recombinant DNA. You can see that this is circular, this is double-stranded. Some part has been displayed in orange, or some part has been displayed in blue. It means this recombinant DNA, which is circular, which is double-stranded, it has been created by joining two different pieces of DNA. So it can be called as a hybrid DNA. One piece has been isolated from one organism and the other piece has been isolated from any other organism, and both the pieces have been joined together to make a single hybrid DNA, and that hybrid DNA is known as recombinant DNA. So to understand the concept of recombinant DNA further, we can concentrate on this diagram in which it has been displayed. What has been displayed? It is a construction of recombinant DNA from human and bacterial DNA. See, there are two organisms. One is human and other one is bacterium. One DNA is coming from human and one DNA is coming from bacterium, and they both are being joined to produce what kind of DNA? That we are talking about, that is recombinant DNA. So how it is being done? Let's see. This is the human cell and this one is the bacterial cell. Human cell is having nucleus, and inside nucleus there is all the DNA. And this DNA is also known as the genome. You might have remembered the genome. This bacterial cell is containing two different types of DNA molecules. One is known as the plasmid, which is circular, which is double-stranded, which is capable of autonomous replication. And another DNA is known as the bacterial chromosome. You might have studied in class 11th about these. So what is being happening here is, first we isolate the DNA from the human cell. In the same way, we isolate the plasmid DNA from the bacterial cell. Now we are having intact plasmid at one hand and all genome of human cell at the other hand. Now what we do, we cut the genome of the human from here and from here, so that this particular segment of DNA comes out, and that particular segment is basically a gene. In the same way, we cut the plasmid of we cut the plasmid of bacterial cell at a single point, so that the plasmid has now been opened, and that plasmid is known as the cut plasmid. Earlier it was intact, it was perfectly circular, it was covalently closed circle, but now that plasmid has been opened. Then what happens? What we do? We allow that gene which is coming from human, we allow that gene to join with the cut plasmid. And if they join with each other, they give rise to what DNA? They give rise to recombinant DNA. Why? Because this particular part is coming from a human cell. It is the gene of a human being. And this particular part, this pink one, dark pink part has been coming from the plasmid of a bacterial cell. So this DNA has two parts, one corresponding to human and the other one corresponding to bacterium. That's why it can be called as a recombinant DNA. Why it is called recombinant DNA? You know that it is a common prefix. It means again. Recombinant means something which has been produced by combination. So in this particular process, if you see what we have done, we have isolated a DNA from here and another DNA from here, and we have combined both the DNA to again produce a single unit of DNA. So we are cutting the DNA and we are recombining the DNA. That's why this particular product, this particular type of DNA, which is known as recombinant DNA. So RDT is basically concerned with the construction of recombinant DNA, and this entire process is artificial. It takes place inside glasswares, inside artificial conditions of the laboratory. The first ever RDNA was produced by Herbert W. Boyer and Stanley and Cohen in 1970s, and they performed their first successful recombinant DNA experiment using bacterial genes. That's why they both, Cohen at the left and Mr. Boyer at the right, they both are known as the father of recombinant DNA technology. What they did, they isolated antibiotic resistance genes from the plasmid of Escherichia coli. It is a kind of bacterium, you know very well. And they ligated, they joined these genes to the plasmid of another bacterium named as Salmonella typhimurium. You might have heard about Salmonella. It is a kind of bacterium. So what they did, they isolated the antibiotic resistance genes from one bacterium, that is E. coli, and they joined these genes to the plasmid of another bacterium, that is Salmonella, and by this joining they produce what kind of DNA? They produce the first ever recombinant DNA. So accordingly, the origin of this recombinant DNA technology is because of these two scientists, Stanley Cohen and Herbert Boyer. So RDT is basically, what is RDT now? We can say that RDT is basically concerned with the construction of recombinant DNA. Now, the second most important part of RDT is use of that rDNA for generating some kind of human welfare. How it can be used, how it can generate human welfare. The techniques are highly diverse. The techniques are highly differ from one point to another. And these techniques we will study, we will discuss through our lectures successively. We will go into deep details of the same. Till then, that's all for today. Thanks for watching, and queries are welcome. Okay, take care, bye.